what they call nowadays transposable and transposed elements. And yeah, this became in fact the topic of my studies over the last couple of years, let's say the last 10 years. Anyway, I learned that, yeah, I saw and I learned that we are the product of intelligence, of intelligent design. And this is also going to be the topic of my talk today: learning processes, plasticity of the brain, and how these transposable and transposed elements are involved in it. Okay, so if you want to know something in general, then you need to ask questions and you have to look for answers on these questions. And that's exactly what we are going to do today. We are going to address a few questions. First of all, what is learning? Then second, what is variation? How is variation induced in genomes? And it is important because in plastic phenomena like learning processes, variation is a very important factor. And we will see that transposable and transposed elements are an intricate part of the genome that is able to induce this variation from within. And then finally, we will have a look at what TEs are and how they are involved in learning processes. This is rather new. So first of all, we have to look into what is learning. And I think everybody knows what learning is, because you sit, take a book and try to grasp what is in this book, or you try to, for instance, in sports, you wield a bat and try to improve your tactics and so forth in sports. This is all processes of learning. And if we look into the most accessed platform for information nowadays, Wikipedia, we see that learning is the process of acquiring new understanding, knowledge, behaviors, skills, values, attitudes, and preferences. Well, okay, this is a very broad definition, of course, because you can learn, in fact, everything you like. But if you look deeper and further, you go further, then you also find more biological definitions like this one. Learning is a physical process in which new knowledge and skills are represented by new brain cell connections. And the strength and formation of these connections are facilitated by chemicals in the brain, which we call growth factors. So this is, in fact, how we see learning processes nowadays in the scientific paradigm as improved and rearranged neuronal connections in the brain. And you can also visualize learning processes. This is, for instance, a functional mRNA image. And this shows the increased activity of brain regions, which is demonstrated by increased blood, increased blood flow. And here in this panel on the left-hand side, you can see that the blue color indicates active regions or active areas in the brain with increased blood flow when people start to learn a new task. And you can see that also the blood flow decreases when this task is already mastered. And eventually, when you have skills, learned this new task, you see that different areas of the brain take over, in fact, these new tasks when you master them. So you see that the brain is rather plastic here, and that different locations of the brain are actually involved in learning and in mastering the task. So already here you see plasticity of the brain. And if we look into the functional units of the brain who are involved in these learning processes, we see here the neurons. And this is, of course, an artist's impression of an electrical signal, which can be seen as this very reddish, what is now a yellow spot. On the left-hand side, I don't have a cursor, unfortunately, but you can see it. And it's an electrical signal that is shooting down the nerve cell and then off to the others in the brain. And learning strengthens these pathways that these signals take, essentially wiring certain common pathways through the brain. And here on the right-hand inset, this is a neuron from a mouse brain, which shows the cell body. As you can see, this is the whitish dot on the left. And you can also see the axon projecting from it. And as the brain learns, the neurons relay information faster and more efficiently. And in fact, this mouse was genetically modified to make a fluorescence protein that glows green under a microscope, fluorescent microscope. And you can then certainly see these axons and the body of the neurons. Well, in the next slide, we see a scheme of this learning unit, the neuron, and it is clear that it is divided, in fact, in two parts. You see on the left-hand side, you see the soma, or the cell body, and this cell body contains also the nucleus, where the DNA is located, which acts as a control center for the whole cell. And what you also see in the soma is these dendrites. And this is a spike-like structure which makes contact with the so-called terminal button here on the right-hand side. And this is already part of what we call the axon. And the axon is in fact a very long, very thin cable that is conducting the signals, the electrical signals. And it should be noted that these myelin sheaths that are wrapped around the axon come actually from glial cells. And this is the orange cell here in the middle of this frame. And it's a spider-like cell, and it is called glial cells. And these cells make up the major part of the brain. About 85% of the brain is made of glial cells. And they are very important also in learning processes because they wrap themselves around these axon sheaths. And it's a bit like a blanket, and they form then this myelin sheath, and this myelin is made of protein and fatty acids and insulates the axons. And this insulation prevents electrical signals from inappropriate leaking out of one wire, what we call the axon here, and into another. So it is very important that you are conducting the signals in the axon and that there is no leakage. Myelin is a bit like, let's say, the plastic that is coating the jackets of copper wires in electrical devices, you can say. And as you can see, they have these tentacles and they wrap themselves around the myelin sheath. And in the brain, it is like this, that about every neuron is then accompanied by 4 to 5 glial cells that keep really the axon insulated. And this speeds up the rate of neural signaling and also helps to prevent confusion, crosstalk between neighboring nerve cells. So they really have a very, very important function. And several studies also found that when new skills are learned, the amount of myelin insulating and around the axon is increasing. Sometimes even not new glial cells are formed and they wrap themselves around naked axons that are being formed by these neurons. And so the increase of the myelin sheath is a very important part of the learning processes. There's also another role for these myelin sheaths because it actually speeds up the electrical signal along the axon. And that is because the glial cells force the signal, in fact, to jump from the one cleft to the other. And so the signal from the start to the end of the axon is increasing. And in this way, the signal moves faster than when it was simply a small tube. You can say it's like flying from one edge, from one cleft to the other, instead of just traveling all along this axon. So there are several mechanisms in the brain to improve the learning process and to the signaling in the neurons. And it's not only the glial cells that have a very important role, but also, of course, the connections that the neurons make. And that is then visualized here in this slide. You get more dendrites. The somatic bodies produce more dendrites, so they can also make more connections with the terminal ends of the axons. And these terminal ends connections with the dendrites are actually called the synapses. So addition of more neurons or neuronal synapses and interactions between these neuron cells increases the neural circuitry involving more and more neurons. So the connections and synapses increase and they form together these neural circuitry and stabilizes then the processes that are leading to learning. Of course, all ins and outs are not known, but the physiology, how these learning processes are thought to be formed are rather well described. So in conclusion, we can say here that what is learning? It is, in fact, if we simply look at the physiology, it is an increase of the myelin sheath around the axons so that signaling in the brain is improved. And also there's an increase and rearrangement of neural connections. And this is called as a rewiring, or as synaptic plasticity of the brain, it is also sometimes called. And this is the way we think currently about learning processes. How it is involved in higher processes such as consciousness and so forth, it is currently completely not clear. There are many more things that play a role there, but in learning processes, we think that these two processes are the most important ones. And if we talk about plasticity, we also talk about variation, and variation has been one of my topics of research for a very long time. And we will address two sub-questions here. How is variation generated in genomes? And when and where is variation required? Well, we go back a long time in history, then we end up with one of the first persons who studied variation a lot. And this is, of course, Charles Darwin, everybody recognized him. And he says in his 1859 book, The Origin of Species, he said, "I have hitherto sometimes spoken as if variation had been due to chance. This, of course, is wholly incorrect expression, but it serves to acknowledge plainly our ignorance of the cause of each particular variation. In those days, it was completely unknown where variation stems from. It was simply taken as a fact that there is variation in nature." If we go further if we look further into what Darwin wrote on this type of things, he later wrote in 1871 that the birth of the species, which is actually due to variation and speciation processes, he says then, and of the individual are equally parts of this grand sequence of events which our minds refuse to accept as the result of blind chance. So then he thinks, or he started to believe, that the variation that we observe and which leads to these observations like speciation is due to blind chance. Well, variation is very pivotal to adaptation. That's what we know. And even to speciation. We also know that that organisms can split up and become different species, but the term variation itself is very vague. It's not very well defined. And the question is variation at all self-evident? That is also a good question to be asked. And nowadays it is so that if we look into what variation is and biological variation, we end up also again at this information platform. And here it is mentioned that variation in biology usually refers to the heritable differences within a species. This is important in genetics and the study of evolution in natural populations. Heritable variation is ultimately caused by mutations of genes and chromosomes and shuffled by recombination. So this is in fact biological information, and biological information is driving these sort of processes. So the current idea is that variation is generated by mutations. And if we talk about mutations in the evolutionary paradigm, these are copying errors, just plain errors in genetic code that afterwards can be selected. Well, these sort of processes cannot play a role in variation in the brain, for instance, because there is nothing to select, because neurons do not divide, and selection can only take place when there is an entity that is able to reproduce. And if they don't do that, they simply are unable to be selected. So I said, really, is it really copying errors? There must be something else. And I was actually triggered, or I actually very excited by this particular experiment that was carried out early 2000 and presented in Science a couple of years later. And it is an experiment of Susan Estes, and she was a PhD student in Spain in those days, and she did experiments with populations of C. elegans, and that is the roundworm, which is often used in genetic studies. And what she wanted to know is something about diversity. How is diversity reformed after you send these roundworms through a bottleneck? And this is actually what she observed. She cultured these worms, then she reduced the population to very, very few organisms. And she was actually puzzled to find that she had, after a few generations, after the bottleneck, the diversity was as great as before the bottleneck. And in fact, she would have expected this, that there would be for a very long time less diversity, less variation in this population, because she severely restricted the genetics, the gene pool in this experiment. So she was quite puzzled by these results. And in those days, I already started to study transposable elements. So I immediately thought, okay, there must be some type of variation-inducing mechanism present in the genomes that are actively generating these type of variation. And I, if I remember correctly, I even sent an email to her to inform her about these transposable elements in the genome that could explain her peculiar results, because she found it rather peculiar. And of course, they are rather peculiar, because if we are only dealing with point mutations and errors in genetic code, they are hard to explain. So this hints already to a mechanism in the genome that is actively generating variation. So how is variation generated? Do organisms have to wait for mutations to occur? No, I think it is much more elegant. And in the meantime, I think this has been shown beyond any doubt in many papers in the literature that genomes generate variation themselves. They have variation-inducing genetic mechanisms. And this is also clear from one of the most important, I think, experiments of the last 20, 30 years, is the so-called Lansky experiment. And I studied in the early 2000 years, I studied these experiments quite intensively, because I was also very intrigued in how bacteria, which he cultured for years and years and years, were able to change over time. And what he actually did, he cultured these, and he's still culturing actually these organisms, these bacteria, E. coli. He cultured them in those days for 15 years, week after week, and he had about 20,000 generations. And the thing is, that is 20,000 generations of these bacteria is about 400 trillion bacteria that were present in this experiment. And what he did, he cultured a week and another week and another week. And eventually he presented the data in a few papers around 2004, which, as said, I studied. And what we can say about these experiments, that there is a change of the bacteria. And what we see is that the reproduction rate increased by about 70% in these populations. There's also a loss of genetic programs. That was an abundant that was abundantly observed in these studies, and it is in accord, of course, with the use it or lose it rule in biology. If you don't use certain genetic programs, you are going to lose them. And also the major changes that he established during the first years, and thereafter there were only minor changes. And also what is intriguing, I think, that separately evolved strains acquired the same or similar gene regulatory patterns, and it was assessed with gene arrays. But I think one of the most important findings was that genetic changes were predominantly done by transposable DNA elements. And we know these sequences as so-called insertion sequences, and it's also known as an IS element, and IS elements are transposons. So in bacteria also, there is definite proof, evidence, that changes, genetic changes, can be driven and predominantly are driven by transposable elements. And the fact is, what are these elements doing? They induce variation in offspring. It is a sort of search program to search for novel genetic contacts. And genetic elements, these genetic elements have the ability to translocate, to transpose, and cause genetic diversity in bacteria. And the interesting part is that we all know about such genetic elements, at least if you are in genetics like me and other geneticists, we all knew about these elements. And all organisms, they have them, and they were in those days still regarded as junk. But these elements are transposons, or nowadays they are called transposable and transposed elements. So every organism has them, and we know this already a very long time, and they've always been seen as selfish DNA or junk DNA, but they were already discovered in the 1940s, 50s by Barbara McClintock. And in those days, people find it so peculiar because they were certain about it that genomes were rather static. And every now and then they changed a bit, and then they could be selected over millions of years. But what she found is that the genome is very plastic. There are elements in the genome that actively rearrange the genome. And as said, it was completely ignored because people did not understand the findings. And then in the 80s, it was in fact rediscovered. End of the 70s, it was rediscovered, mostly by Roy Britton, I think, one of these geneticists from the United States. And it was so obvious that she was right, so she gave her the Nobel Prize in 1983 for her discoveries. Well, as said, they were long regarded as junk DNA, and in my book of Albert's Molecular Biology of Cell, it is still shows a sentence that they are DNA sequences with properties that ensure their own retention in the genome, but which do nothing to help the survival of the cells containing them. Well, this is now completely outdated. It's in 20 years, it is completely outdated, this view, because these so-called junk DNA are now fulfilling a lot of genomic and genetic functions. And this is a very simple example how they, for instance, are able to generate a variation in species. If you have, for instance, plants like mice, what was studied by Barbara McClintock, you have here a pigment gene that is able to generate a color. And if a transposon jumps into it, the gene is interrupted and the color disappears. And the result is things like this. And this is exactly what Barbara McClintock studied, these type of things. And she found, due to her studies, that these transposons do the work. Another intriguing example, of course, was recently described is the Bettelaria moth in England, very famous for industrial melanism and always presented as a school book example of natural selection or evolution, but it is also driven by a transposable element. So this also shows the importance of these elements in variation. And this is a figure that I published a few years ago in the Journal of Creation. Here we see what VIGs, I call them VIGs, variation-inducing genetic elements. Officially, they are known as TEs, transposable elements. But okay, what can these elements do in the genome? First of all, they're able to facilitate chromosomal recombinations, and I will show later how this works. And they are thus arranging karyotypes. And the arrangement of karyotypes is, of course, the same as inducing variation. And this eventually could even lead to speciation because the karyotypes of the different organisms are no longer compatible and they cannot reproduce together anymore. On the other hand, and this is shown on the bottom of this panel, VIGs are also involved, or TEs are also involved in duplication and translocation. They easily translocate, they easily duplicate, and in the presence of genes, they regulate the genetic output. And also, if you vary the genetic output, this is also known as variation. And this can easily lead also to adaptations. And many adaptations in nature have already been linked to these, the effects of transposable elements. Well, what are TEs? I already mentioned that they are transposons and that we find them in all organisms that we have studied. I already mentioned the IS elements in bacteria. They're very important in generation of variation, but also in plants, in yeast, in insects, we all find transposons. And we do not find a few types, but we find hundreds different of types of transposons in all sorts of organisms. In plants, for instance, we see transposons, and they have they code for genes that help to transpose to another position in the genome, thereby generating the variation. In yeast and in insects, you have the TI and copia elements, and they are more and more complex because these transposons require an RNA intermediate. So it is not just a simple jumping around system. It is an intricate system that is also controlled and regulated by the genome itself. And currently we know that in the transposition of, for instance, a line element, which is also found in mammals here, there are about 100 genes of the genome involved to control and regulate these transpositions. Well, here we see the ERV, the endogenous retroviruses, and the lines, and these are found predominantly in the genome of mammals, and they are also found in the genome of humans. And here we see what is present in the genome of the human being. We have about 45% of our genome contains these transposable elements. So this shows already that they are abundantly present. Almost half of the genome is built of these transposable elements. So this already indicates that they must have a very important role in biological processes. And if we look into a classification of these transposable elements, we see here that there are DNA transposons, which is only a minor part of all the transposable elements in our genome. It's only 2.8%. About 300,000 of them are in the genome, and they do not require an RNA intermediate to transpose. They simply transpose as a DNA molecule, which is then cut out of the genome and put in another place in the genome. It's simply a cut and paste mechanism. On the other hand, we see that about 42% of the transposable elements consist of an element that uses an RNA intermediate. It means that it is transcribed, then it is reverse transcribed and put back into the genome. And as such, it is generating also variation. And we have two very well defined transposable elements, RNA with an RNA intermediate, which are the non-LTRs, non-long-terminal repeat elements. They are called lines in science. And on the other hand, we have these ERVs, which do have an LTR, long-terminal repeat. And these are the so-called endogenous retroviruses. I will not go into details here, because if I go into details here, I would need another hour to explain the differences, how they work, etc. We simply have to drive home the message that these elements are mobile elements present in the genome. They're highly controlled, their activity is highly controlled, and they are able to put the genome in novel genetic contexts. And this is nothing else than variation. Well, this is an overview of these TEs. We have a lot of them in our genome. The frog has about 70 to 80%. Mice has a lot. Humans, 43%, 45%. No, and it goes all the way down. The more genetically simple the organism starts, the less of these elements we have. And bacteria only have DNA transposons, and there are not a lot of them, but still they function in the way that I described before. Okay, So, in fact, what are they, these TEs? Well, all eukaryotic genomes are littered, and also prokaryotic genomes, of course, have them. But eukaryotic genomes are really littered with so-called endogenous retroviruses. These are the ERVs and the lines, and in our in the what is it in the normal thinking, there are also remnants of them. They are called signs and long-term repeats. These signs and long-term repeats simply function as genetic switches in epigenetic and genetic processes. Long-term repeats, for instance, are just isolated promoters from which genetic expression can start. And we have really millions of them in the genome. So in fact, our genome is a box, a switch box with genes that are controlled by thousands and thousands of these elements. And of course, you can generate a lot of variation when you vary the elements that are involved in these processes. Okay, as said, nowadays they are known as transposable and transposed elements, abbreviated TE. So, in fact, we are dealing with transposons, and how do they affect gene expression? That is schematized here. In the middle, we see the transposable element, and depending on where it sits in the genome or where it is located in the genome, it can have different functions. As you see in the yellow panel, they can function as enhancers, and enhancers are in fact driving expression or increasing the expression of genes. They can also function as alternative promoters. Alternative promoters are usually in front of the promoter, but they can also be located in exons, between exons, for instance, in the entrance. Further, they can integrate themselves in the entrance when they are active. They can thus also modulate and start to affect gene expression. One thing that is not very often observed, but we will see that it also plays it plays a role in neurons, is exonization. And exonization is simply that it integrates in an exon, and usually it upsets the gene, and the exon is disturbed and cannot be expressed anymore. So this is also a type of inducing variation by a TE. And finally, and this was recently also described, that TEs can integrate in this 3' untranslated region here on the right-hand side, thereby affecting the RNA stabilization that is transcribed from the gene. These 3' untranslated regions contain usually sequences where microRNA binds that affects the stability of the RNA. So if it integrates there, you also have an effect on the gene expression, because the RNA that is being transcribed is more or less stable, and it can more or less be expressed. So there's several ways to affect the gene expression by transposable elements, and all these mechanisms have also been described. For an overview, you can go to this paper that I wrote a few years ago together with one of my colleagues, European Respiratory Journal. It's also playing roles in diseases, for instance. Anyway, with this mechanism, you can generate a lot of variation, almost infinite variation. And I think this is one of the seminal papers. This is also referring to transposons in the genome. It was published 11 years ago in Nature Genetics, and it is called The Regulated Retrotransposon Transcriptome of Mammalian Cells. And what they showed is, or what they conclude is that retrotransposons, and then we are talking about these lines and these ERVs, the retrotransposon transcription has a key influence upon the transcriptional output of the mammalian genome. Many genes start from these retrotransposons and not from the promoters, as we would expect from our theories of the past. So up to 30 to 50% of the genome, of the genes of the genome, start from these so-called endogenous retrovirus-like elements. Well, what are TEs? What are they good at? I already told you that they are very good at inducing variation. And this has been shown over and over the years. In early 1990, the late 1990s, it was already clear that these transposons can activate cryptic genetic programs in bacteria, that they could induce fitter phenotypes in E. coli, and that they regulate gene expression in yeast. They can also induce chromosomal rearrangements, adaptive mutations, evolvability, for instance, via repressor inactivation. And also, one of the observations is that they are involved in integration processes, for instance, plasmids in bacteria, due to recombination processes on the IS elements, on these genetic elements. And also in plants, we see these mechanisms active. We see that they control epigenetically the genes of Arabidopsis. These are then associated with also these retrotransposons. Well, we can go, in fact, on and on and on. And more papers appear in this field that show that the variation in plants is mainly due to the activity of transposable elements. And I think two interesting examples are also shown here. The morning glory flower pigmentation is regulated by a mule retrotransposon, of course. And another very intriguing example is, of course, the reds and the white grapes, which is also a result of the GRET1 retrotransposon. So in this way, you generate variation in genomes. Good. This is another one, recently published. Transposable elements drive rapid phenotypic variation in this one, in the shepherd's purse. It's just to show that most of the variation that we, for instance, observe here in flowering is due to transposable elements. So the natural variation in nature that we observe is not just a random process, but we have mechanisms for that can be induced. All right. So what are TEs? Well, I renamed them 10 to 15 years ago as variation-inducing genetic elements, because this is in fact the function that they have in the genome. They are inducing, and they can induce very rapidly, genetic variation. So you do not have to wait for mutations to occur over thousands of years. No, you have an active mechanism in your genome that is able to do this. TEs are tissue-specific epigenetic regulators of gene expression and cellular responses. And TEs are mobile genetic elements that populate genomes where the host largely controls activity. And this activity is controlled by for instance, by C methylation in epigenetic responses, but there are also very important pathways in our genome, the so-called SI pathway and the pRNA pathway that actively controls the activity of these transposons. So it's all regulated and controlled in biology, and it is very important. And currently, they are no longer regarded a priori as junk DNA, but they still are regarded as the remnants of viruses. And if you have read something about my articles in the Journal of Creation, you know that I have also proposed that viruses, certain viruses like the retroviruses, are in fact modified transposable elements, which are known as the endogenous retroviruses. And the endogenous retrovirus, which is a transposon-like element, already looks so much like an RNA virus, a retrovirus, that it only has to add 1 or 2 genes to its genome to become a full-blown RNA virus. So I think that the origin of viruses can also be found in these type of retrotransposons of our genome. Good. So we have very, very much evidence that genetic variation is predominantly due to the activity of transposable elements that are already present in genomes. And I think we should rename these elements as 2 variation-inducing genetic elements. And if you want to know more about it, you can go to these 4 papers that I wrote over time, where I explain how these things work, how viruses can emerge from these TEs and so forth. Good. How then are these TEs involved in learning? Well, we already have shown, and this is what I showed you before, that these TEs are very good at generating variation in genomes. And they do this by duplication, translocation, by facilitating chromosomal rearrangements, and so forth. And in this way, they are able to generate the variation, which could lead also to adaptations. And in fact, learning is adaptation of the brain. This is what we see. It's a rearrangement of neurons. It's a rearrangement of connections between neurons. So it is not far-fetched to think that these transposable elements have also an important role in adaptation of the brain in learning processes, because learning is in fact rearrangement of the neuronal connections, which is known as neuronal rewiring, and variation is in fact rearrangement of genetic connections, which is also known as genetic rewiring. So question, 1 + 1 is 3, 1 + 2 is 3, I mean, could learning be related to variation of genetic connections in neuronal cells? And I was not even very surprised to find that indeed transposons are active in neurons of the fruit fly, Drosophila, and may there introduce genetic variability. This was the first study, 2005, that showed this, and it was a very intriguing study. And in those days, people did not really know how to interpret these data. So a lot of studies was then redirected into this, and a lot of studies in Drosophila showed indeed and confirmed these variability in the neurons induced by L1. L1 we have already discussed, line 1. This is one of these transposable elements that we find in all organisms, all eukaryotic organisms, I have to say. And then in 2011, there's another, I think, very seminal study that showed that transposons are active in neurons of humans and may also introduce genetic variability. Then a few years later, line 1 activity, one of these retrotransposons, were found to be active in neurons of the hippocampus and in the cortex, and they resulted there in variable regions in the genome, indicating that indeed we are dealing with a sort of variation-inducing mechanism in the neurons. And this is one of these experiments that was carried out in those days. And what they did is they did single-cell retrotransposon capture sequencing. What they do is, in fact, they sequence individual cells of the brain cells, of hippocampal neurons and the glia cells, as well as cortical neurons. And they showed an average of about 14 novel line insertions here. And these must be novel insertions, and they are in very specific places also. And specifically, they were transcribed in neural stem cell enhancers and in hippocampus genes. And this is indeed, this made indeed these researchers suspicious that they must have some sort of roles in learning processes. And in Drosophila and in mice, for instance, they found that it was related to a greater variety of cognitive and behavioral phenotypes. So these neuronal this differentiation in neurons appears to also be driven by variation inducing genetic elements that we call lines, line 1 in this case. Well, here are 2 more studies in 2016, where line in healthy neurons helps to insert and remove stretches of DNA. And line 1, for instance, deletes entire genes, and the consequences are that they cause variation in the neurons. This is also what they started to deduce from these experiments, that these transposable elements are actually actively involved in recreating or rearranging the genes and the genetics of the neurons. And they also reported that about 50% of the neurons are different. So of all these billion neurons that we have in our brain, also have billions of different neurons. Genetically speaking, they are not the same, they are different. And this is due because of these variation-inducing mechanisms in the neurons. In 2018, there was a report that somatic line insertions are found in about 1 per 2 neurons, and even higher number in dentate gyrus granulocyte neurons. This means that the process is not just random, because they compared it to the glial cells, and the glial cells do not have these phenomena. So it is specific for these neuronal cells, and it has a meaning, and the meaning is that it generates variation in these neurons. And you can, of course, understand this because if it is like that the neurons are not the same in your brain, that you require billions of different neurons in these neuron processes, you cannot program this into your genome. Your genome would simply be too big to program all these neurons, all the genetics of these neurons. So what you have to program in is a mechanism that is able to generate this variation. And it is exactly this what the line is doing in your brains. It helps to differentiate your neurons in these billions of different learning types of novel genomes of these neurons. Well, the most recent study I could find is this one is from 2020, where they show that transposons are even spliced into neuronal genes and then potentially change the structure and function of these proteins of the brain. Well, this is, of course, highly peculiar, because normally if there is a transposon that integrates in a gene, it upsets this gene or this exon, and it cannot be expressed anymore. But here we see that it is controlled and highly specifically integrated in these genes, so that the function changes of these proteins in the brain. So this must be a highly controlled and highly regulated process, of which we only start to scratch on the surface. Okay, and what they also were able to show is that many of these genes altered by transposons, by these integrations, have previously been shown to play a role in a broad range of behavior and learning processes. So indeed, it is not just randomly jumping around of these genetic elements, but it is a process that is directed, controlled by the neurons themselves in the DNA. And it makes also sense because we have several mechanisms that control it. Hundreds of genes are involved that control the hopping around of these transposable elements. And it really looks like a design system to me. Okay, so finally, they argued that artificially induced changes of these genes, often induced by transposable elements, can lead to changes of behavior in animal models. So this is the state of the art currently. We see that these so-called junk DNA, which is no longer regarded nowadays as junk DNA, plays a very important role in learning processes in the brain. All ins and outs are not yet clear, but a lot of people started to work and look into this direction. Anyway, to sum it up a bit, these transposable elements generate novel epigenetic context. And I will also not go into detail here because I do not have a lot of time anymore left. But what we see is that there are affecting genetic mechanisms by direct integration in the genome. And on the other hand, this direct integration in the genome is able to affect the epigenetic marks. And both, of course, are related to gene expression and differential gene expression. And we know differential gene expression is nothing else but variation. So TEs regulate gene expression and they alter phenotypes. Emergent findings demonstrate that TEs can establish and rewire gene regulatory networks and circuitry by genetic and epigenetic mechanisms. So it is a highly complicated system, a highly mobile and highly plastic system to generate novel genetic context. And how do they do it? We already saw this. Here we can go through briefly again. They can act as enhancers, as alternative promoters. They internalize, they externalize, and they affect RNA stability. So 5 possible mechanisms already when they integrate in the gene. There's another one. Here we see how, for instance, epigenetic mechanisms could be involved. Here we see a TE. The TE translocates to another position, and the methylation ME patterns change, and programs are being closed down, and other programs can be switched on. So here we have direct effects on the DNA expression levels at the level of epigenetics and genetics. And here we see how we have to understand nowadays the genome. Here is a stretch in the genome. You see there are only a few coding areas, and there is a sea of TEs surrounding them. Every coding area, every exon has several hundreds of these TEs in their neighborhood that are potentially regulating the output of gene expression. And this is then the results. They regulate gene expression, they regulate the pathways, the biological pathways, and thus they regulate the biological processes. So today, a gene is no longer understood as just a stretch of linear DNA, but it is understood as a coding island in an ocean of regulating and controlling gene elements. And most TEs accumulate in this ocean, grouping themselves between the islands, thus changing the genetic or epigenetic context, and so they also change the output of the genes. So in fact, the genome of higher organisms loaded with these TEs is a switch box. So, and this brings me almost to the end of my talk. How are TEs involved in, for instance, facilitation of indel mutations? We have seen that they can remove parts of the genome, thereby creating novel genetic landscapes. And if we start to look into these lines have the same sequences, and they are located throughout the genome. And they are presented here by the R1, R2, R3. And as you can see in the top image, if the recombination takes place on the same line, nothing happens to the genetic context, as you can see. But if you have unequal, for instance, unequal lining up of these lines, and it is here represented by R1 lines up with R2, and this often happens in the genome, you can imagine that you have a duplication or a deletion, which is presented then in the lower panel of this panel. And in a similar way, you can also lose parts of your genome. Genes that you, for instance, do not need in these neurons can be simply deleted. And it is not just a matter of chance deletion, no, it is regulated and controlled, as said before, and it's regulated and controlled by several hundreds of proteins and RNA sequences. But you can also expect extended deletions by crossing overs on lines that are separated by another 100 of lines, for instance. And this whole region then disappears, and you can understand when there are exons or genes, they will also disappear. So this is a very handy and very rapid mechanism to get rid of genetic programs that you do not need in these neurons, for instance. So this brings me to my final slide. How are TEs involved in learning? I think we now start to understand how they are involved in learning by creating and making novel genetic contexts, by making the very specific neurons that are required in these processes. And they do this by a similar mechanism that I described before for variation inducing for organisms. These TEs, they simply are very good at duplication and transposition, which affects genetic outputs, which affects variation, which creates variation, I have to say. And this then leads to adaptation of the brain of several different thousands, millions, billions, I have to say, different neurons. And this is, in my opinion, how the brain creates these different genetic contexts. So there is genetic plasticity possible. Learning is, in fact, adaptation of the brain. And this brings me to the end. If you want to read more how variation is induced in the genome by transposable elements, then I would recommend my book, which was released a few years ago, Darwin Revisited, or how to understand biology in the 21st century. And this is my organization I work for, the association Wort und Wissen. And we study this type of things. We study biology, we study creation, and we praise and give praise to the Lord who created all these intricate systems. I thank you very much for your attention. This is the end of my presentation. Thank you very much for your attention, and I thank God for science.